Okay, so let's talk about northern, western, and southern blotting. So to start off, blotting is a really useful tool for molecular biologists in that it aids in detecting certain proteins, DNA or RNA strands. And this is helpful because it's like using a magnet to help you find a needle in a haystack. Blotting sorts the macromolecules by size or shape, therefore removing what you don't need and making it easier to find what you're looking for. Blotting is especially useful in the medical community because of its role in diagnostic testing. It's also used in a wide range of biology concentrations from biochemistry, farming, ecology, genetics, and beyond. So, Edward Southern was the first scientist to publish about southern blotting, which is why the technique was named after him. He started it in 1975 and used it to look at specific DNA sequences. The other two main types of blotting, western and northern, were named as a homage to Edward Southern. The science community decided to stick with directions when finding names for the blotting techniques. So, just some background info on macromolecules and why we study them. There are a number of macromolecules in biology. The three big ones are RNA, DNA, and proteins. Nucleotides, I included those because they're also very important. They are the backbone of the DNA molecule. And studying these macromolecules and structures and being able to isolate them is helpful when you are studying molecular biology. So just a real-world example of how blotting is useful and applicable. So they don't do this anymore. It's quite outdated, but they used to use blotting as an HIV confirmation test using Western blotting. So after a serum from the patient was collected, the proteins from the cells that are HIV positive would be run through gel electrophoresis, which we'll talk about in a minute, and then they would be washed by the Western blot technique. Now, Western blotting employs antibodies so that the proteins have something to bind to. So after a first round of normal antibodies are used to wash the proteins, the patient's serum would be used as a secondary wash. That way, if any of the antibodies from the patient's serum stick to the proteins, the pathologist would know for sure that they are HIV positive because only those specific antibodies bind to proteins from HIV positive cells, which means that if their body's producing those antibodies, they are definitely HIV positive. So again, there are big three main types of blotting are the southern blot, northern blot, and western blot. A helpful way to remember what each one does is this little word rhyme or called snow. I'll talk about that in just a second. So all three types test for particular macromolecules. Southern blotting detects isolated pieces of DNA. Northern blotting looks for RNA sequences, and Western blotting identifies proteins. So remember this using the mnemonic device SNOW. So the S stands for Southern, the N stands for Northern, or the NO, and the W stands for Western. We know that DNA is made up of RNA, which is made up of proteins. So you know it goes Southern, Northern, Western. Southern tests for DNA, Northern tests for RNA, and Western tests for proteins. Okay, so the Western blot is a pretty common test when looking for specific proteins and trying to confirm a diagnosis. It allows for protein separation by either size or folding. Two examples of using Western blotting would be the HIV confirmation test, as we previously mentioned, and another kind of old school test that they used to use it for was confirming Creutzfeldt-Jakob disease, or it's more commonly known as mad cow disease. CJD disease causes prions to form in the brain tissue, and because prions are irregularly folded proteins that can transfer the shape onto normal proteins, this is how it spreads throughout the brain. A Western blot test can identify these misshaped proteins by catching them because of their irregular folding in size, and you can confirm the diagnosis. So, because Western blotting separates proteins by size or molecular weight, we need to use a gel that is porous, that has holes in it, so we can sift out the proteins from the other macromolecules in our cellular matrix. So, the proteins we usually use will come from inside of a cell that has been lysed or opened. Proteins have to first be denatured before they can be used for blotting because of their differing charges. So by denaturing them, we force all the proteins we're looking at to be negatively charged, and therefore they will want to travel to the positive electrode and onto the membrane, which is our end goal with Western blotting. So let's talk about the SDS page common setup for a Western blot, and SDS page stands for SDS polyacrylamide gel electrophoresis. So this is a standard setup that you would use to perform a Western blot. You would need a buffer tank, electrical charge, your electrode assembly, or your and a gel holder, also called like a cassette case. You'd also need a buffer solution. You would need a power source, some filter paper and membrane, because again it's a blot, pipettes, and probably something to keep it cool. It depends on how much you're running and for how long. There are three main steps for Western blot: separate, transfer, and detect. So let's get started for our hypothetical Western blot here. So to start, we need to prepare our protein sample. We would denature it using controlled heat and then add it to some diluted buffer. Then we will place our gel in the cassette case and put that inside the tank. After filling the tank with buffer, we would load the wells of our gel with our protein sample with a pipette, and we would run the gel at a certain voltage. So after a predetermined amount of time, we would take the cassette out and make the quote-unquote sandwich in the order listed here: sponge, filter paper, gel, blotting membrane, filter paper, sponge, which again is shown over here. You want to make sure to keep everything wet. The sandwich would then go back into the cassette cases and then into the tank, with the membrane facing the positive side, again, because our charge on our proteins is going to be negative, so we want them to run to the positive side. So after running our sample at the appropriate voltage, we must accommodate for the antibodies that we need to bind with the proteins we're studying. But if we just throw antibodies at our membrane, they will bind with the membrane and not our proteins. So the first thing we do is we add something called a blocking buffer, like milk, yes, just like normal over-the-counter milk, or something called BSA. And you would incubate the membrane in the blocking buffer for a specified amount of time at usually 4 degrees Celsius. So what this does is it kind of keeps the antibodies from just binding to the protein and not to our proteins, but binding to the membrane. What it'll do is that initial one will bind to those blocking buffer. So after you incubate and you do a wash, you're going to get rid of the excess blocking buffer, and then you're going to incubate the membrane again, but this time with secondary antibody solution. And to help the secondary antibodies bind with the primary antibodies, something called a reporter enzyme is used. This enzyme helps the secondary antibodies stack up on a protein, which makes that protein more visible on the membrane. we can observe the frequency and distribution of our protein in our sample. Two other kinds of secondary antibodies are common, and it's usually radioactive labeling or fluorescent dye. So now we've run our gel, we've washed it, we've incubated it. Now we can finally look at our results. We can analyze our proteins on our membrane. We can look at frequency, size, and comparison to the standard. So what you usually do is you run a standard DNA ladder on one side of your gel, and that'll just be something for you to compare the rest of your samples to. So on here you can see all the different dark and light colored bands are the different protein pieces that you would be looking at. And you can look at their size, their range, their frequency, and analyze your results. So another technique is called northern blotting. It's similar to Western in that it uses electrophoresis, but instead of looking at what we looked at with the Western blot, we're going to look at RNA fragments. And to do this, we use something called a capillary system for molecule transfer. So instead of using the sandwich like it did with the Western blot, the gel will be placed on a membrane in a buffer solution, and capillary action will transfer the fragments. And instead of just enzymes used to bind the RNA fragments, there are also probes that are made of nucleic acids and complementary to the well, hold on, folks. Having some technical difficulties here on my end. Alright, apologies for that. So these probes, they are complementary to RNA strands. These probes, like the enzymes, help the RNA fragment stand out on the membrane for observation. So here's our tools and procedures for our northern blot. Pretty much the same electrophoresis setup again, like a Western blot. Except now we're going to use a formaldehyde wash for the controlled breakdown, and that's kind of going to work like our incubating buffer we used in our Western blot. So we're going to run the gel, we're going to add our RNA fragments to the wells, and we're going to use the ladder as a control again. After we ran our gel, however, we're going to place it in a transfer buffer, and we're going to add capillary blotting paper on top with some paper towels and a weight. And what that's going to do is that's going to transfer our molecules onto the blotting paper via capillary action. And then we're going to add the probes and wash our membrane with a solution that will ensure our probe attachment. And then we can actually detect these probe signals on X-ray film. So a kind of real-world example or application of RNA or looking at RNA via a northern blot is it allows us to notice change across organ systems, developmental stages, and medical treatments. We can also use northern blotting for cancer monitoring, in that we can check for changes in tumor suppression genes and cells. So last but not least is the southern blot. Again, uses very similar techniques to the other two. This is used to look at DNA fragments. Gel electrophoresis is used as before. The original blotting technique is southern blotting, again, named after Edward Southern, who created it in 1975. And here's just kind of like a little diagram of how it goes. So here's your materials you would need. Gel electrophoresis setup, same as before, a buffer solution, the same nylon membrane, a capillary transfer system, or if you're old school, you can do a vacuum system as well, and hybridization probes. And so what we would do first is we would obtain our DNA fragments, which would be cut into fragments by endonucleases, and then we would run it through electrophoresis gel. Then